the massive metagenomic data analysis using abundance-based machine learning. And after this last talk, I hope that all of us will just move to the poster session close to the posters by the Meta sub, and we'll just continue discussion there. Okay, thank you.
Hi, everyone. Thanks for sticking around. Like they said, my name is Zach. I'm from St. Louis University, and today I'm going to be talking about massive metagenomic data analysis using abundance-based machine learning. So just to give a spoiler, this talk is going to be profoundly similar to most of the talks we've seen today. So just keep that in mind. So the last two speakers really set up this problem quite nicely. So I'm not going to spend too much time talking about the data set itself. But I will say that we did interpret this challenge as having two very specific questions. The first one is, can we predict a city label from a sample that we don't know? And then second, if we have a set of samples that we've never seen before, can we predict the number of cities that exist within that data set or that set of samples? So, to answer both of these questions, we're going to take the same general approach, where we're going to start with some kind of data quality control using a tool like bbduck. We're going to do some kind of a metagenome assembly using something like megahit. We're going to approximate abundance using something like bbmap. And then finally, we're going to make some kind of prediction using some kind of machine learning model. But what I think we kind of add to this, and what I think is probably a better title for this talk, is that a reduced representation data set is still able to sufficiently predict city labels. So this is necessitated because St. Louis University doesn't have what we would call fantastic computational resources, but we do okay, so we kind of just did what we could do. Specifically, we wanted to test a reduced representation approach using two very specific paradigms. So in the first paradigm, we're going to maintain the integrity of the paired-in data structure, but we're only going to take half the samples from each of the cities. And then in the second paradigm, we're going to get rid of that paired-in data structure, taking only the first or the left read from each sample, but we're going to take it from each sample from each city. So the rest of this talk is essentially just comparing these two methods and looking at what happens. So a reasonable place to start, of course, is how the assembly went. It should probably be a surprise to no one that the paired-in assembly took a little bit longer, but it generated what looks to be like a less fragmented assembly. But in both cases, we generated about 10 million contigs. Now, we can do machine learning with 10 million features, but we probably don't particularly want to. So we wanted to find some kind of approach to filter this data set down to make the models more manageable. So what we ended up choosing to do was filtering based on length of the sequences, and we just wanted to find lengths that would normalize the mapping rate if we took the raw reads and aligned those back to the metagenome assembly. So we just wanted that to be roughly equal in both of the paradigms that we go with. So what that looks like then is in the paired-in paradigm, we have 242,000 features, and in the left-only paradigm, we have 2 million features. But these models should be comparable in the end. So a reasonable first question would be, is there any structure in this data set that exists without using any kind of supervised methodology? And a great way to visualize that is to look in principal component space. So if we look using the paired-in paradigm, we see two clear clusters, but we can't really draw any profound conclusions from that. Using the left-only paradigm, we had a couple outliers on PC1, so we can't really draw anything from that. Another reasonable question, of course, is what if we use some kind of a supervised approach? Can we see clear structure in the data? And it turns out a good way to do this is looking at linear discriminant space. So when we do this and we look at these abundances in linear discriminant space, we see a really nice structure. We can see clear clusters are present within this data set. So that gives us a lot of confidence that if we do some kind of supervised machine learning approach, we're probably going to be able to generate a reasonable accuracy. So like many others before me, we just kind of did a random walk through scikit-learn, choosing models that have slightly different underlying assumptions, and slightly different underlying mathematics. We took the models that tended to perform the best, which, as we've seen before, tend to be simpler models like random forests, and we explore those to try to find what generally performs the best. So specifically in both of the paradigms, we looked at a linear discriminant analysis, a random forest with 10 estimators, and a naive Bayes. And these are the tenfold cross-validation ranges. And these are heat maps of the confusion matrices. The light blues aren't showing up. But in general, what we see is the strong diagonal, since it's not normalized, it doesn't look as strong, just generally means that the models are performing relatively well. What we get out of this, though, is that the random forest in both of the implementations tends to perform a little bit better, so we specifically look at the random forest. So here, what we're looking at is essentially, we're trying to tune the number of estimators. So we want to know what the optimal number of estimators or the optimal number of decision trees going into the model would be. So here we're looking at random iterations of test train splits across random segregations of decision tree building. So the general conclusion we get from this is that so long as we have about 20 estimators in both of the models, we tend to get pretty good accuracy. So we chose a number right in the middle. We chose 50. And in both of these implementations, then, we're able to achieve about a 90% accuracy with these models. And again, these are heat maps of the confusion matrices where the strong diagonal shows that we're able to perform relatively well. Just a quick interjection then. Let's say for some reason the 242,000 or the 2 million features that we're dealing with you don't want to deal with. You want to reduce this data set even further. You can do that if you employ something like binning, which is a popular metagenomic tool. So specifically what we did was we used two different implementations of maxbin, and we were able to reduce from 242,000 or 2 million features down to either 220 or 260 features, depending on the parameters we used for maxbin. This can be discussed later if anyone's interested. We collapsed all of the different bins into an eigenspecies, and then we used the transformations in that abundance space to predict unknown samples. Like I said, we can talk more about that later. But using the random forest with 50 estimators, we can get about an 83% accuracy. So it's not performing as well because we're obviously losing some information, but it's still doing okay. So we have a set of final predictions for that first round of unknown data. Unfortunately, someone might be me, didn't email someone back, so we don't know the true labels. But we would love to talk about that with people that do have the true labels. I can tell you just off the top of my head, it doesn't look like we performed particularly well. None of my models tended to predict New York, and OFA was very underpredicted. So then that leads us to this final question, which is, can we predict the number of cities given a set of labelless data? And this is an interesting question, but it's particularly challenging. And we can tell by looking at the principal component space where there's no clear structure that segregates the cities in this space. Obviously, we could do something like manifold learning and maybe get something out of it, but as it stands, it doesn't look particularly good. So we kind of came up with this approach just to see what would happen, where we take the labeled cities abundances, so this first release of the data, and we iterate over a reasonable number of clusters that we would expect, which in this first data set was 8. So we kind of go up below and above 8. We do unsupervised clustering using a few different implementations, and then after each calculation, we'll run some kind of a post-cluster metric, something like a Kolinsky-Harabash score. And then if we do this in a bootstrapped fashion, we can aggregate these post-cluster metrics, and hopefully what we would see is a strong peak over the true number of labels, which in this case we know as being 8. Theoretically, that would happen. Unfortunately, what we tend to see is a peak a little bit below 8, so we see it around 6 or 7, and then we see another really strong peak start to come out at about 2 times K, so we're still trying to untangle what exactly is happening there. So then just general conclusions. It seems, insofar as the data we have, that this is a fairly valid approach, this reduced representation. Obviously, we would want to test this on a few more programs. Megahit isn't the answer to the universe. So we want to try a few more things. And then again, kind of something that we've gone back to several times, this is absolutely devoid of any biological context, so we haven't dug in at all to what this actually means. I'd like to thank anyone that funded any member of our team, and especially St. Louis University's Bioinformatics and Computational Biology program, because they took me in and I'm not really one of them. So thanks.
Thanks a lot. We will just have